All right, here's our 9th video, Living Environment Regents. We're talking about transcription today. We're talking about protein synthesis and especially transcription. So in the next video, we'll talk about translation. They go together, right? But we're breaking them down slowly. So basically in transcription, and we'll get our pen going in here. In transcription, the body, DNA will form RNA. And RNA, and specifically now for this video, we're talking about mRNA, messenger RNA. Messenger RNA will basically code your genes, code your gene, and it will leave the nucleus. It'll leave the nucleus and it will find the ribosome and it will start making proteins. That's basically what it does. But in this part, we're going to talk about how the body makes mRNA, what mRNA does before it leaves the nucleus. That'll be transcription. So let's make our double helix, right? There it is, right? So you can see it has nitrogenous bases, the A, T, G, and C, which we talked about in the last video. Adenine or adenine, some people say adenine, cytosine, cytosine, thymine, guanine, however you want to pronounce it. And don't forget, let's get our pen. Where is our pen? We lost our pen. There's also, we're going to talk about is uracil. And I talked about this in the last video also, because mRNA is going to take from adenine, it's going to, instead of making thymine, it's going to make uracil. And one of the reasons is outside the nucleus, thymine will not last. It will not last outside the nucleus because of oxygen. All right, so let's get a better picture. Well, let's draw our, I call them these sine waves. They look like sine waves. We're drawing and then let me use a different color for the other strand. So that's our one strand, and then we have another strand, right? And here's our nitrogenous bases, or nucleotide bases, whatever you want to call them, and they're all bonded by hydrogen bonds. So again, if let's say we have, let's just make A, T, C, C. Let's try that. So in DNA, A will bond with thymine, and it will have a bond here, and there are two hydrogen bonds, not bombs, for adenine and thymine. Now, and T will bond with A, and cytosine with guanine, it has, this bond will have three. There'll be three hydrogen bonds. Three hydrogen bonds. All right. And of course, C to G. Let's do another one over here just to make sure you understand what they bond to. What would this one become? A becomes T, right? C to G, and T to A. All right. That's in DNA, but when RNA goes through, it'll bond it differently. And we'll talk about that in a minute. So we have our DNA, and for teaching purposes, I'm just going to make looking like a ladder. And we'll have, we have to have some way of this breaking apart. So we're going to break this apart. It has to break the bonds. And in order for mRNA to form, to code, it needs these bonds broken. And that's when we're going to talk about this thing called an RNA. I'll write it over here. An RNA polymerase. RNA polymerase will basically attach themselves to a promoter, and the promoter and the mRNA polymerase will unwind the DNA, so it will code. So an RNA molecule is formed, and then it will code it. So let's look at how it does that. Okay, let's change the color up. Let's simplify this. So we have our DNA. We have a strand on top. It's not necessarily on top. We have a strand that you see on top and this one you see on the bottom. And there's names for them. So we have a coding strand. We'll say coding strand, which also has another name. It's also called the sense strand. So you might hear it as the the sense strand or the coding strand. Now the bottom one is called the template strand. And that's the one that mRNA will code. That's the template strand. And it's also called the antisense. So I guess when something doesn't make sense is antisense, logically, right? Just using a little joke there. So, and it's not really top or bottom because you know how the double helix is. Let's draw the double helix again. So we'll have this, let's say this is the coding strand. See how it, goes up and down like a roller coaster. And then we'll have our template strand. So you can see it goes up and down like a roller coaster. Now the coding strand is going from 5 prime. We talked about 5 prime in the last video to 3 prime. And on the opposite end, it goes from 3 prime to 5 prime. So when I talked about the mRNA, the RNA polymerase, they will code from the 3 prime to the 5 prime so they can create mRNA going from 5 prime to 3 prime. Just a basic review again with 5 prime and 3 prime. Remember, in DNA, and we'll just say that's our backbone of DNA, right? These are the nitrogenous bases, right? There is a pentose sugar, because it's like a pentagon, right? We have a pentagon here. This is the pentose sugar, which is the ribose, the diribose in DNA. And these circles are carbons, right? So there are 5 carbon molecules, right? This is the first, this is the second, the third. The first bonds with the nitrogenous bases, let's say adenine, for example. And this 5th carbon over here, there's the 4th one, will bond with phosphate. Because remember, DNA is phosphate sugar backbone. And when you see, and let's get a picture here so you can see. And this was in the last video also. All right, so here's the 5N. You can see on the 5N, we have our phosphate, but on the opposite end, you have the hydrogen-oxygen, hydroxyl. It ends, the 5N ends with the hydroxyl at the 3 end, and the opposite of that is the phosphate. You can see it's upside down. So that's what they mean by 5N and the 3 end. So for the RNA polymerase, they will code going from the 3 end up into the 5 end. And when they code, they'll create an mRNA that's going from the 5 end to the 3 end. And we'll look at that. So the mRNA polymerase, they broke apart. Let's get our pen going again. They broke apart DNA at the promoter, right? The promoter. Will we fit it in? And here it is. Let's just say it's this thing like this, right? Being oversimplified. So it's going from the 3 end to the 5 end. And again, that is the template strand. So this will be the coding strand. And it will take that, whatever it is, and it will create mRNA. So let's get a better picture. Let's try to draw a little more neater. That's DNA replication. And we'll talk about that in other videos coming up. All right, now let's get our pen. That's what I want. Get our little pen going here. So again, it broke apart the hydrogen bonds. And let's do a couple of those over there. Now they're broken apart. For example, and let's just say that over here we had A, C, C, G, G, and let's say a T. So on the bottom of the DNA, this would have been T, this would have been G, G, C, C, A, and we have an extra one here, right? Well, let's just say that's T, and we'll make another one, unless I missed one, A. Fix that up there, A. So again, this is the anti-sense strand. This is the sense strand. This is the coding strand. And this is the template strand. So and we'll make another color here. We'll say this is the polymerase, RNA polymerase going through and to code. So when it codes, this T then will become an A. The G will become a C. C, G, G. Now the A is not going to become T. It's going to become a U. U for uracil, because RNA will not code for thymine, because when thymine basically can't leave the nucleus. As I said, oxygen will destroy it. And the T will become A. So this is our new mRNA created from the RNA polymerase. As you can see, it's making the exact copy of the sense strand right there. And so then this mRNA, which is now formed, will leave the nucleus. Let's see it leaving the nucleus. Get a picture of that. Here it is, leaving the nucleus, and it will go into the cytoplasm, which we're going to talk about this in translation. It'll find the ribosome, and then we'll talk about tRNA, amino acids, and all the rest. All right, so that is our video for today. Great job.